First of all, of course, thank you very much for giving me the opportunity to talk. The title of my talk is what I actually initially wanted to talk about, doing a lot of advertisement for our stuff, and then I reread the assignment, which was, please answer these questions. So I came up with a couple of suggestions, answers I want to discuss in more detail in what follows. First, I think we have a problem of the availability of data, of very specific data for quantitative modeling. There is this saying, build the road and they will come. And I think we as modelers have frequently gotten this the wrong way around. We thought if we build computational models that can do all kinds of fancy things, then experimental biologists will just flock around our knees throwing data at us, and usually it doesn't happen. So I think what we need, and really as part of this project, is to think about how we can improve this situation, that most of the currently available experimental data lack the temporal and spatial resolution we actually need to build and validate detailed mechanistic models of cellular signaling pathways. And this, in many cases, leads to the situation where people say, okay, for the sake of simplicity, I do this and this and this. And they develop models and perform simulations, and then there is a visual inspection, basically. The results of some visual experimental data are compared to visual simulation outputs, and the measure of quality is whether it looks similar, and I think we need to do much better. And this means also that we need standardized experimental protocols in terms of cell type, condition, stimuli, especially if we want to combine data from different sources. And then, obviously, to make the transition from the data to the modeling, we need methods to encode spatial-temporal experimental data. I will talk about this in a bit more detail in a moment, and also about this, how to develop tools for computational modeling in realistic and even dynamic models of cellular morphology. One of the challenges I see is that we need to develop tools for efficient computational modeling across different scales. Many people have said this before. Obviously, at the fundamental level, everything that happens inside of the cell is stochastic, but we can obviously not perform fully particle-based stochastic simulations of entire cells. So we have to come up with hybrid approaches, and this is a grand challenge, I think. I want to comment on this a bit. The necessity to make underlying assumptions of computational models fully accessible to the most often biological experimental experts, because having a model and performing simulations of it really only makes sense if you can convince the experts, the biological experts, that what you're doing actually makes sense. And I want to discuss this challenge with the example of EGF receptor signaling. Here you have a typical diagram showing at the top the EGF receptor interacting with all kinds of components. And a very important question here is which interactions between the receptor and its binding partners can be simultaneous and which others are competitive. And yes, you can encode all the details in a differential equation model, but don't try to explain this to an experimental biologist, even though this is fully valid and extremely important. We need to find a way to convey the information to experimental people. And there is this fantastic work from the groups from Edwards and Wilson in Albuquerque. And they used a combination of molecular modeling and simulation to explore what kinds of interactions between the EGF receptor and downstream components can happen simultaneously and which others are competitive. And they came up with this table summarizing the results. Now, in the end, to simulate this quantitatively, they had to convert it again into these kinds of differential equations, which obviously are not readily accessible to experimental biologists. So I want to focus just on this interaction between Schick and the phosphotyrosine 1148. So if you look at this set of rules, basically describing who can bind simultaneously with Schick, then grasping this immediately is difficult. We developed a graphical method of encoding molecular interactions that, once you've defined these interactions, automatically is translated into the equivalent of partial differential equations and can be used even for simulations where you have intricate cellular morphologies and even dynamic cellular morphologies. And here, this set of rules looks relatively simple. We have one, you see the red dotted tag, which encodes whether PLC gamma is bound. And if you look very closely, you see it doesn't matter. So for this interaction, we can set it to don't care. There is one crucial condition: PLC gamma must not be bound to the lower domain of the receptor. And we can encode this graphically. And then after the binding, one of the other tags keeping track of binding records that, yes, Schick is bound. And as I said, you can define the interactions graphically, and then in the end, it's translated automatically into quantitative simulations. I next want to come to what I think is an underappreciated problem in the simulation, the spatial simulation of cellular behavior, namely that if you have a cellular signaling pathway and you describe the signaling pathway without the spatial component, it may look very simple. Yeah, for example, this one. There are two receptors, receptor 1, R1 and R2, and they can interact with components in cell 1 and cell 2. For example, C1, when it binds to R1, the complex can then bind to the receptor 2 on cell 2, and so on. A very simple network. But if you translate it into a simulation where you actually have two cells adjacent interacting with each other, then the whole situation becomes very difficult, because reaction number 2 creates a complex that is visible in 5 different spatial compartments. So you need to represent the third from the top, the third complex. You need to represent it in cytoplasm 1, membrane 1, intercellular space, membrane 2, and cytoplasm 2. And computationally, this is very difficult. Let me give you an extreme example. The interaction between adhesion receptors that can bind in trans between two cells and in cis on the surface of one cell. The resulting network is extremely simple if you don't take space into account. If you take space into account, it becomes a nightmare. And I think here we need approaches that translate definitions of molecular interactions automatically and even in the context of dynamic cell morphologies into simulations. And that means that no, the partial differential equations cannot be defined at model definition time, but they also cannot be defined at compile time. They have to be generated through the simulation at runtime, basically. And then we can populate, for example, POTS model systems of cells with these receptors. And then if there is an extension of the POTS model volume elements, the software can then, during runtime, regenerate the necessary cellular signaling networks and take care of all the necessary biochemical details. And then you can, for example, perform simulations like this one, where cells are moving and interacting. And importantly, here the cells really change much of their intracellular signaling networks when they make a contact. So this is not an animation or phenomenological description of the receptor interaction networks, but a full biochemical simulation. One of the questions was, how do we build a real and enduring community? And I want to say what we need, and I know this is contrary to what was suggested by Phil. We need to define a driving biological problem with well-specified goals, and we can discuss this later. And again, the same answer for the question, how can such a community be most effective? I mean, Phil provided an answer through new software paradigms, akin to the app model, and so on. And I think, yes, those are all valid points, but I think we really should define a driving biological problem with problem areas on various scales that can be tackled by specific groups, as well as obviously interface approaches to link those partial solutions. And one suggestion I would like to make is, or what we could look at is the macrophage data set that was collected by the Alliance for Cell Signaling. Again, that data set at the moment doesn't have the necessary spatial temporal resolution, but we could build on it. And this is where I want to end. Thank you.